So what we just discussed, right, LTD mediated by AMPA receptor internalization, that's what we see in the Schaefer collateral pathway, right, the CA3 to CA1 neurons in the hippocampus. But there's another place that we see LTD, and we've mentioned this one before, right? We can see LTD within the cerebellum, specifically in what are called Purkinje neurons. And the cerebellum here. That's supposed to be cerebellum. I got a little loose with it. All right. So I think one thing that's important to mention here is that there are two other types of synaptic inputs coming in to these really extensive dendrites of the Purkinje neurons, right? So the Purkinje neurons have these enormous arborizations, these enormous dendrites. And so these Purkinje fibers are receiving synaptic input, and it's excitatory. From two sources. The first would be climbing fibers, and these are in no particular order. So climbing fibers and what are called parallel fibers. Okay, so both are going to be important here. Okay, so the parallel fibers are axons and axon terminals of these granule cells. And climbing fibers are ones that are axons of actual what are called climbing fiber cells. Then we've got our Purkinje cells right here. Okay, so those are the three different neurons that are going to be involved in this circuit. The second thing I think that's really important is that this is only seen when climbing fibers and parallel fibers are active at the same time. So they must be active simultaneously. Right, this, we know what a word is for this. This is associativity. Right, if you go back to when we were first talking about mechanisms of LTP, we saw associativity. How does this work? Right, and this is where we'll go through it relatively quickly, because again, we've talked through this once already. We know that AMPA receptors are the ones that lead to depolarization when glutamate is present, right? So initially, right, the parallel fiber, so this is where we're starting, right, with the parallel fiber, releases glutamate. That glutamate binds AMPA receptors postsynaptically. So we'll say AMPA receptor mediated depolarization. Okay. But also occurring at the same time, and maybe more importantly, for the long-term portion of this long-term depression, is that metabotropic glutamate receptors, those G protein coupled glutamate receptors, mGluRs are active. Right? Because they also bind glutamate. And this initiates the signal transduction cascade that leads to diacylglycerol and IP3. Right? So the result of this, right, through PIP2 and phospholipase C, is an increased concentration of diacylglycerol as well as IP3. Okay, so that's what's going on in the parallel fiber. What about in the climbing fiber? The climbing fiber also releases glutamate, and it binds to receptors slightly away from the dendritic spine, like right near the dendritic spine. And so this activates ligand-gated calcium channels. So the glutamate binds and ligand-gated calcium channels open up. Hopefully this looks a little bit familiar. And calcium then comes in the cell, right? So calcium enters the Purkinje neuron. So now we've got optimum conditions here for the activation of PKC, right? PKC needs increased calcium and diacylglycerol. Right? And these two molecules go a long way to activate PKC. And what PKC does is it's able to phosphorylate its targets, and through a mechanism that's not exactly known, right? PKC triggers the internalization of AMPA receptors. Okay. Again, we think that this is endocytosis in a clathrin-dependent manner, right? So receptor-mediated endocytosis, meaning that there has to be a specific protein near the target of internalization, right, that functions as a receptor to determine what gets internalized. Otherwise, right, internalization might bring in the mGlu-Rs, which is not what we want to do, right? So what gets internalized are these AMPA receptors due to the targets, right, clathrin, serving as the signal for where internalization will occur. Okay, so you can see in this diagram what we just talked about, right, that the glutamate, yes, it activates these AMPA receptors initially, but it also activates metabotropic or G protein coupled glutamate receptors. This gives us diacylglycerol and some level of calcium increase. But the calcium, really, we need higher levels of calcium than that to activate PKC. That calcium comes from the climbing fiber synapse. Right, so calcium goes up, and these two factors converge on PKC. The result is, right, and again, we don't know exactly how this happens, but PKC phosphorylates some substrate that triggers the internalization of AMPA. Okay, the general schematic is up here, right, where climbing fibers and parallel fibers are bringing their impulse to the axon terminal at the same time, right? And through this mechanism, we can get a weakened synapse. So let's take a look at the data right here. So we're measuring the parallel fiber EPSP, okay? And so what this is going to show is we're going to stimulate, oops, hang on for 1 second. I'm tied up in my microphone here. Okay, sorry about that. You'd think I'd have this wireless mic, but I don't. I got a cord that is hanging off this headset that I'm wearing as I record these. So what I want to do is just show you the data, right? In this experiment, we're going to record from the Purkinje neuron when we stimulate the parallel fiber. And the parallel fiber is stimulated. And so now we're seeing some EPSP, right? And under control conditions, when you stimulate parallel fiber, the response that we get, this EPSP, is what, 6 millivolts, and then we do it again, and then there's one that's maybe right about 6 again, and then we do it again, and we get one that's maybe a little less, right? So we're right around 6.1, 6.2 millivolts. That's what a typical EPSP is that's measured in the Purkinje neuron when the parallel fiber is stimulated, okay? But then the Purkinje fiber, that's what excuse me, the parallel fiber, that's what this is, right? So we're going to pair the stimulus in the parallel fiber and the climbing fiber, right? So if we look up at A, we're going to stimulate here, and I don't know if they have the other one here. Where's the parallel fiber stimulus? And here, right? Somewhere on the parallel fiber at the same time. And we're going to record, right, from the Purkinje fiber. And what we can see is after we pair the stimulus between the parallel fiber and the climbing fiber, is then when we go back to just the stimulus from the parallel fiber, right? So we only need to do this climbing fiber stimulus once, right? So now this is going to be back to only parallel fiber stimulus. You can see a substantial reduction for a long time in the EPSP as measured from the Purkinje neuron, right? So if normally that EPSP that measured in the Purkinje neuron would be 6 millivolts, right? We stimulate the parallel fiber and we see the EPSP in the Purkinje neuron is 6 millivolts. Now, right out at 80 minutes when we do that, stimulate the parallel fiber, after these things have been paired, after the stimuli have been paired, now the Purkinje neuron responds with only a less than 4 millivolt EPSP, right? So it's still going toward threshold, right, this EPSP, but its magnitude is reduced, okay? There's one more type of plasticity that I want to talk about, right? We've talked about LTP, LTD in mammals, but I want to talk about something called spike timing dependent plasticity. And this will wrap us up for this chapter. Spike timing dependent plasticity. I missed one. Spike timing dependent plasticity. Okay, STDP. And look, at this point we've talked about plasticity with respect to how rates of stimulus are what influences plasticity, right? So with either LTP or LTD, right, it was all about the rate of stimulus, right? Mattered. That was the big determinant of LTP or LTD. How quickly or slowly do we stimulate? Okay, but this type of plasticity, spike timing dependent plasticity, doesn't rely on the frequency of stimulus. Instead, it depends on the relationship of temporal activity. I'll explain what I mean in a second. When you see this word temporal, what do you think? Hopefully you think time, right? The temporal relationship. Of activity between the pre and postsynaptic cell. So I'll say of pre and post synaptic activity. Okay, so I think the best way to talk about this would be to look at the figures, and maybe we'll do that in this last video for our day today.